Welcome to my world. Another day, another time, another period for knowledge acquisition. Knowledge is power, health is wealth. Today, I'm going more into the technical importance of laboratory bottles color. Everybody that comes to the lab or have one test or the other to do in the lab will likely collect a bottle, a sample bottle that you find that they have colors, especially the cover, the lid that covers that bottle is colored and you find various colors on them. But because you are so used to the same color, probably you never uh, checked the importance or know the importance. But this importance is also very necessary in helping make sure that the result you are getting is exactly what you are expected to get, because there is what we call the pre-analytical stage of error, where if the samples are not collected properly in the right bottle, then there is bound to be the wrong result. So when you are involved in knowing what the right bottles are for samples collection, you have that confidence, because knowledge will always give you confidence that what is being done as far as your test is concerned is the appropriate thing. So let's go on to the slide section. But like I've always said, if you like what you are listening to, press the like button, subscribe, and let this continue. Importance of lab bottles colors. The outline: objective, introduction, need for perfection, advent of Vacutina, different colors of bottles, and the outcome. Objective: to expose the reason for color differentiation of laboratory bottles, awaken their interest, and also promote the collaboration of stakeholders, be it patient or medical practitioner, for a reliable results. The introduction: the exact period blood collection started is unknown, but the need for blood collection is clear, because blood has to be analyzed and you have to take a little sample. But in 1947, Joseph invented the modern glass vacuum after the glass bottles that we were using for cross-matching and grouping. And by 1947, Becton, Dictin, and Company later changed and trademarked the name Vacutina. We used commonly the plastic bottles for a long while before the Becton Dictin Research Center developed this Vacutina Plus. Being in use now, it is a vacuum tube. The vacuum pressure ensures the correct volume of blood enters the tube from the needle, so it is a perfect system. The further introduction: the reason for blood collection, like I earlier said, is in a bottle, is in order to analyze the content of the blood or its components, which are in 2 or 3 parts: the red blood cells, the liquid part called plasma or serum. This is the solid part that is at the bottom, and the liquid part is that golden color, which is either plasma. Need for perfection can be understood that the old method, we were just more or less inject using venipuncture, use a syringe and needle, collect the blood, put it into these bottles. There could be hemolysis during the transfer from the syringes, and there is also that imperfect ratio of mixture, incorrect venipuncture, and standardization is seriously a challenge. Different colors. Why we need the different colors is because the hematological primary investigation estimate the blood cell component of whole blood, and so the investigation requires whole blood collection. So you can imagine that you have to have whole blood. Then the blood transfusion needs clotted sample, so you must use the serum for your serology and what have you. The chemical analysis check extracellular values of enzymes and other substances usually present in the blood, outside the cells. And then the anticoagulant, the fourth reason, the anticoagulant that will interfere with the analysis, thereby necessitating using other alternative. The purple blood bottle contain EDTA, which acts as a potent anticoagulant by binding to calcium in the blood. EDTA really helps to do all these full blood counts, erythrocytes, sedimentation rate. The pink bottle performs almost the same way as the purple bottles, and is also used for transfusion. The blue bottle is used for hematology tests involving the clotting system, which requires inactivating whole blood for analysis. It contains buffer sodium citrate, which acts as a reversible coagulant. The test performed, as I said, is coagulation screening, bleeding time. We have the gold yellow bottle. These bottles are used for a wide range tests requiring separate serum for analysis, including biochemistry, endocrinology, oncology, toxicology, microbiology, and immunology. It is known in the lab as a serum separating tube. It contains two agents, silica particles and a serum separating gel. The silica particle works to activate clotting and cause the blood cells to clump together. The serum separator consists of an inert polymer gel which floats as a layer between the blood cell and the plasma to form a physical barrier between them. The sample can be centrifuged in the lab and the separated serum easily removed. That's the good thing about it. The pre-analytical phase error are many, and you need to understand this reason why we just have to improve standardization. The reasons are incorrect venipuncture, which I had earlier mentioned, the hemolysis of the red blood cells, collection into the wrong containers because you might use the wrong, and instability of some chemical substances in whole blood serum and plasma. These are the errors commonly found. These are the bottles color and what the test they do. The blue bottle, like I earlier said, for coagulation screening. The dimer, the gold color for both chemistry and lipid profile, all the function. The purple, full blood count, hematological. The gray has that sodium fluoride, which helps for glycolysis, glucose estimation. These are all bottles that are of different colors. And then finally, the outcome. The Vacutainer has reduced the error traceable to the pre-analytical stage, thereby improving on the reliability and productivity of laboratory results. It is now a recommended standard for research work because everything is standardized, allowing for quality output. When you use this, you get more. Please tune on to more of my channels to receive more on this.